Now, we've touched on some aspects of recombinant DNA technology already when we spoke of the use of bacteria in laboratory settings, particularly with respect to things like conjugation and transformation, and when we discussed how information could be inserted into bacterial cells. When we talk about recombinant DNA technology, recombinant being the operating word, and that is referring to creating new combinations of genetic material using genetic information from different species. And so here in this simple illustration, you can see that a gene responsible for coding for the human insulin protein, or one subunit of it, can be stuck into a plasmid of a bacterium. Now, recombinant DNA is formed in the lab by sticking together DNA from two sources. It's a complicated process, but it's actually quite easy to do in the lab. And if um, we're in the lab, it's something that we could do even as a lab exercise. Although the success rate's generally quite low in labs, so I've stopped doing that years ago in genetics labs, given the time we have. But it is a very straightforward process once you understand exactly what's going on. Okay? So, one of the most important materials that we use are naturally occurring enzymes called restriction enzymes, or restriction endonucleases, more precisely. And these enzymes are produced by bacteria and they protect them against viral infections. So basically, what these bacteria are doing is they're producing enzymes that float around in their cells and are looking for double stranded genetic material, presumably stuff that shouldn't be there, and they attack it and chop it up. Okay? Now, restriction endonucleases recognize a very specific DNA sequence. So each enzyme will recognize only a certain sequence of DNA and cut that sequence of DNA in a very specific way. Okay? And when it does this, it may or may not leave overhanging ends. And in the next couple slides, that will become very clear what I mean by that. But it's never random how it cuts in that spot. It cuts in the same spot all the time, and it leaves the same aftermath, whether that's overhanging ends or blunt ends. And so the chunks that you're left with after exposing DNA to a restriction enzyme are restriction fragments. So they're fragments of DNA that have been digested with a restriction enzyme. So here's a very simple example of a double-stranded DNA molecule that, you can imagine it extends far to the left and far to the right, but what we're focusing on right now is this 6 base pair stretch of GAATC that is recognized by the restriction enzyme EcoR1. Okay? And most restriction enzymes are named after the organism from which they're derived. So this one is from Escherichia coli, or E. coli, as you've probably learned of it before. That's where the ECO comes from. Okay? So after treating DNA, whether it's bacterial DNA, viral DNA, your DNA, with this enzyme, EcoR1, it will cut every time it encounters this 6 nucleotide sequence in the DNA. It will cut it. It will cut it between the G and the T on the top left and the bottom right. Okay? And you'll see what it leaves are these 2 overhanging ends. The right strand has a 5' overhang, as does the left strand, but it has a 5' overhang on the right side. You see what I'm saying? So they have complementary tails or overhangs. Sometimes these are called sticky ends, because if the enzyme cuts it and goes away, it's quite possible these ends will just come back together. Okay? So that stickiness is useful in a laboratory setting. Okay. Now, here's 4 more restriction enzymes from 4 different bacterial species that recognize very different regions of a DNA sequence. You can see that Hindi3 at the top recognizes a 6 nucleotide sequence. BAMH1 also recognizes a 6 nucleotide sequence. But SAO3A down at the bottom cuts at a 4 nucleotide sequence. Okay? And so the result of these cuts is highly dependent on what enzyme you're working with. And so let's first look at the sticky ends created by SAO3A down at the bottom. Okay, you see that there's a 4 nucleotide overhang on each of those double-stranded molecules. But look at ALU1. That is an example of a restriction enzyme that leaves blunt ends. There is no sticky overhang. There's no tail hanging off. Okay, so these are just the ways in which restriction enzymes work. And there are hundreds of restriction enzymes that have been identified. And the vast majority of these, well, I'm sorry, I'd say slightly less than half of them, you can actually order. So when I need restriction enzymes, and I've used lots and lots and lots, thousands and thousands of dollars worth of restriction enzymes in the lab, you just go on the website of a restriction enzyme company and you order what you want. Okay, you might order basically a quarter of a teaspoon for anywhere from $60 to $800, and you get it overnight the next day. They grow these enzymes, they produce them using bacteria, as you might imagine, and they sell them commercially for quite a bit of money. So anyway, these are very useful enzymes. They're very easy to access, and using them in the way that I'm going to describe is incredibly powerful for genetic research. Now, one thing that is important to think about when you're using restriction enzymes, we're not really going to talk about the implications of this any further, but it's important to realize that these enzymes are not cutting the genome randomly. They cut the genome only where they find the specific sequence that they recognize. And so that sequence might occur a lot in a genome if it's a very short sequence like SAO3A down there, or it might occur less frequently because it's a specific 6 nucleotide sequence. So just by chance, you would expect GATC to occur in the genome, that 4 nucleotide sequence, more often than the 6 nucleotide sequence of AAGCTT that Hindi3 recognizes. Because there are only 4 possible nucleotides, these actually occur pretty often just by chance. Okay, so let's figure out the probability of a SAO3A1 cut site. Okay, if you pick a random 4 nucleotide region of the genome, what's the probability that the first nucleotide is a G? Well, it's 0.25. What's the probability that the second nucleotide is an A? 0.25, and then a T, 0.25, and then a C, 0.25. Okay, the probability of finding that sequence specifically is 0.25 to the 4th. Okay, that is a very small number. Okay, so that's not going to occur all that often, but you know what occurs even less frequently is a 6 nucleotide cut site, such as that recognized by Hindi3. So if you're chopping up the genome, you can chop it up into different sized pieces depending on what enzyme you choose. If you want to chop it up into small pieces, use a enzyme that cuts a 4 nucleotide sequence. If you want to cut it up into really big pieces, pick something that recognizes 6 or 7 or even 8 nucleotide sequences. They will occur much less frequently. Okay. Now, the reason that this is very useful to us in a laboratory setting is because when we recombine genetic information from different organisms, it's made a lot easier if we can just stick them together like Legos. And we can do that if they both have these sticky ends, and the sticky ends that they each have are complementary to one another. Okay? So if I cut 2 samples of DNA with the same enzyme, if I cut your DNA and I cut your cat's DNA with the same enzyme and put them together in the tube, then we can get some of the cat and human DNA to stick together. Now, that's not necessarily useful, but you'll see in a minute how this can actually be quite powerful. So here's an example where we have two different DNA molecules, the green one and the purple one. They are both digested with EcoR1 restriction enzyme, and it leaves complementary overhangs. And those fragments with these sticky or complementary tails can come together. Now we still end up with that gap. So the hydrogen bonds are going to form. That's the sticky part, no problem. Okay? Now the gap that we have here is the exact same gap we get when we get rid of those primers after DNA replication. Remember? Filling in the gaps from those Okazaki fragment primers. The polymerase fills that gap in with nucleotides, but then connecting these last two, it needs a ligase. And so guess what? The company that sells me the restriction enzymes also sells me ligase. I throw that into the tube with my cat and human DNA, and I can glue them both together. Okay? All right. So this is just the basic foundation of how we can create combinations of DNA from different species. This is why the corn that you drive by or the soybeans that you drive by in the agricultural fields in this area, why they have no weeds growing in the yard or in the plot, it's because they're spraying the whole thing with Roundup, and it doesn't kill the crops because the crops have been engineered. They stuck a bacterial gene in the plant, just like this, in a place that allows the plant to make the protein that breaks down Roundup so they don't die. Okay? So, how do we move this DNA where we want it? Okay? Or get it into something that can produce the thing we're looking for. So, insulin would be a great example. Well, we use vectors. Okay, and vectors are basically a mechanism for moving DNA and making lots of it. Okay, so we can put a vector into a bacterium and have that bacterium produce lots of copies of that DNA and or produce lots of copies of the protein that is coded for by that DNA. Okay, so in the case of insulin, what we do is we stick that vector containing the insulin gene into a bacterium, and we just have those bacterium make lots and lots of insulin. That's what we want. So what characteristics are we looking for in a vector? Well, the first is we need a vector that can replicate independently in the host cell, so we can get lots of copies of it even within a single cell. We need it to have restriction sites. We need to be able to cut it so it has sticky ends and we can stick what we want into it. Okay? And then we need it to be selectable. We need to know if a bacterium that we have sampled, whether it's in a tube, a test tube in liquid, or on a plate, we need to know when we see a bacterium growing that it does or does not have the thing that we tried to stick into it. Okay? So we're going to go through all of this in great detail. So here's an example of a plasmid vector. This one is called PUC18. It's simply a bacterial plasmid that is engineered, and we know exactly what's where in this. It's a circular chromosome, much smaller than a bacterial full chromosome, but it's thousands of nucleotides long. Okay, so somewhere in the neighborhood of, let's just call it 2,500 nucleotides all the way around. You can think of it as being something like the F factor we spoke about earlier in the semester. But what's interesting about this vector is that on the left you'll see the only gene, well, the two genes that we care about are colored in purple here. On the left is a gene that confers resistance to antibiotic. So this plasmid, if it is in a bacterium, will give that bacterium immunity to any antibiotic. Well, this antibiotic in particular, ampicillin. The gene on the right lacks Z. Okay, see on the bottom right here, this is the gene that when the cell, a bacterial cell, is exposed to this chemical called XGAL, the bacterium uses it and processes it, and it turns the bacteria blue. Okay, so we've got two genes in here that are important. Make it resistant to antibiotic, and we can make that cell blue. Okay, now in this LAC-Z gene region, in the zoomed out portion, you will see in what is called the polylinker region, there are lots of restriction enzyme cut sites. All of these different things are different restriction enzymes you can use to cut this vector. Why would you cut it? We would cut it because what we're going to do is we are going to stick into that cut site some of the DNA that we're interested in. Okay, that is indicated here by the orange fragments, and you'll notice that they have overhangs on both ends, same as the plasmid that has been cut with our restriction enzyme. The ends of the purple and the orange are complementary. They're sticky with respect to one another because both DNA molecules were cut with a restriction enzyme. Okay, so they get stuck back in. Ligase makes that last 5-3 connection, and we have now a recombinant plasmid, and this plasmid cannot metabolize XGAL. It does not allow a cell that contains it to metabolize XGAL because we stuck something in the middle of that LAC-Z gene. We split it up. It can't be transcribed properly. You can understand how that would now happen, because if you tried to transcribe and translate the LAC-Z gene, there's a whole nother big chunk of something we just stuck in there, and it would screw it all up. So you're not going to get blueness if you have this plasmid. You will get ampicillin resistance. Okay, so this is called bacterial cloning, because what we're going to do is put this into cells, and then we grow a boatload of those cells, and every time that cell divides, it is cloning, making an identical copy of whatever's in these plasmids. So when we grow these things on a plate, we're going to see some colonies that are white and some colonies that are blue. And you should know what that means now. You should know which one of these we want. Think about us putting in the middle of that polylinker region, we put a gene for the protein in insulin. Which of these has that gene stuck in it, the white ones or the blue ones? Okay, well, it's only the ones that have the LAC-Z gene disrupted and are white that have the insulin gene in them. So let's look at a summary here. On the right is the DNA we chop up with restriction enzymes. On the left is our plasmid vector that has been digested with the same restriction enzyme. In the middle, the vector has taken up our target DNA, or our DNA of interest, the purple stuff. And there are three possible outcomes illustrated at the bottom. When we get bacterial cells, we don't just try to put this vector into a single bacterium, right? I have a tube that has thousands of cells that I hope will take up this vector. Competent cells, that's a term that should be familiar to you. I have thousands of competent cells in a tube, and I put a bunch of these vectors that I've cut, and I put a bunch of my DNA that I've cut, and I try to get the DNA to ligate into the plasmid vector, and then I do something. I shock the cells, those competent cells, to try to get them to take up the plasmid. But then there are three options of bacterial cells. On the left, circled in red here, is a cell that received the plasmid, okay? But it did not get the chunk of DNA that I tried to put into it. It didn't get that purple bit, okay? And so what that means is this cell can grow on antibiotic, okay? So if we put ampicillin on our bacterial plate, this bacterium will grow. But what color is it going to be? This bacterium does not have a chunk of DNA stuck in the middle of the LAC-Z gene. It is still able to metabolize XGAL, and it turns blue. So this is one of the blue colonies. That would make a blue colony. This cell has received the vector, the plasmid vector, so it can grow an antibiotic. And that vector it received had a chunk of DNA stuck in the middle of LAC-Z. So it cannot utilize the XGAL. It will not be blue. So this colony is going to be white. And then this cell, this is the third possibility, did not receive a plasmid, can't grow in the presence of antibiotic. We don't ever see it again. Okay? All right. So cloning is a very important and powerful tool that has been used and is still used quite frequently. And it's probably the simplest example of recombinant DNA technology, right? I've cloned DNA from frogs and snakes and lizards and insects into plasmids and had bacteria grow and produce them for me. All right. Now, making lots of copies of a molecule of DNA using a plasmid or a bacterium is helpful. It's more useful for producing lots of proteins as an end product. Now, in order to simply make lots of copies of the DNA, if we're not interested in the protein end product, we would utilize something called the polymerase chain reaction, or PCR. Okay? And so this is a reaction that is done in a tube, and it is damn near the same thing you learned as DNA replication. Okay? But the difference is, in DNA replication, you will recall we are making copies of the entire chromosome. Okay? You're replicating the entire chromosome at one time, and the end product is 2 copies of the same chromosome. In PCR, we are focusing only on one very specific region of the genome. In humans, it would be one gene on one chromosome. That's all we're interested in. Okay? And this is exactly the way that the work that you did in lab number 3 for the Innocence Project, this fragment size analysis. Those fragment sizes are produced, that the sample that is analyzed is the result of a PCR reaction where lots and lots of copies of that little repeat region with its flanking sequence have been made. And in those analyses, you're just figuring out how long is this fragment for this individual. Are they homozygous for it or heterozygous for it? Okay. So in the polymerase chain reaction, much like DNA replication, there has to be a template. You need primers. You need deoxynucleotides, G's, A's, T's, and C's, and you need a polymerase. Those are the same things you need in a cell to replicate DNA. Now, there are 2 differences here. One of which is that the primers are not randomly made. Okay? The randomly made primers that you use in DNA replication, they are necessary to get the process started, and these primers are necessary to get the process started. But unlike DNA replication, these primers are not random. They are very specific. I have to order them from a company, and I tell that company, I need a single-stranded DNA molecule that reads 5' GC GT AC GC GT T 3'. I order a very specific sequence because I know that is what is on one side of the gene region that I want to amplify. And then I give them the sequence, a 20 nucleotide-ish long sequence of bases for another primer I want them to synthesize for me. I order those online. They cost me about $8 a piece. They overnight them to me, and I have these primers that I can use for PCR. So the template is similar. It's identical to DNA replication. It's the original chromosome. If I'm looking at a crime scene sample or I'm doing a cancer genetic test for someone, I'm taking a DNA sample from their cells and extracting DNA, and their DNA is the template. I need polymerase, DNA polymerase, just as we did before. And what I don't need is helicase or gyrase. Okay? All we're doing in this is we are using heat instead of helicase. We are melting those weak hydrogen bonds and breaking apart the double-stranded DNA. Now I'm going to walk through how PCR works for you and then recommend a video. So the way PCR works is it goes through a number of cycles, usually, let's just say, somewhere in the neighborhood of 25 cycles. And remember what your raw materials are in this tube, in your PCR tube. So we have this, we're starting out at the top. We have this double-stranded DNA to be amplified. Let's just say it's a sample from a crime scene. Step one, I heat it up, melt it, denature the DNA. The two strands come apart. Now they're available for primers to bind to them and DNA polymerase to come in and make copies. Okay, so you can see in step two, the primers match and stick in their appropriate regions that they are complementary to. Remember, I had to order very specific primers, so they will only sit here, bottom left, top right. That's the only place in all 23 of your chromosomes, 23 pair. It will only sit on this spot. It'll sit on these spots on the chromosome your mother gave you and that copy of the chromosome your father gave you. But it's only going to sit in that one spot because it's very specific. It will only match that one spot. So the primers anneal, and then DNA polymerase comes in and extends the primers and makes a new strand in blue. Now the one difference in PCR is that that primer is not made of RNA, it's made of DNA. Okay? The reason it had to be RNA in replication is because we needed that 3' end to start making DNA. But because I have a company make it for me, they can synthesize a single-stranded molecule out of DNA without any problem. So now, after step 3, we had 2 copies of that double-stranded molecule. We do another cycle. We heat it up again, melt those 2 double-stranded molecules apart, cool it down, primers bind to it, then we extend. So after 2 cycles, instead of having 1 double-stranded molecule that was the 2 pink strands that you see at the top and the bottom here, now we have 4 double-stranded molecules. Okay? So this process is relatively straightforward if you really understand DNA replication well. The slight differences being the primer is something you buy, it's made of DNA, and it's not random, it sits only in one place in the genome, both of them. And you notice that those 2 primers are on different strands, okay? The 2 primers don't sit on the same strand when that double-stranded molecule splits apart in cycle 1. So it's relatively straightforward, but it's also something people struggle with. It is not uncomplicated, okay? I would recommend Googling PCR explained or searching for a PCR video on YouTube. This one that I've screenshot here has the worst soundtrack possible, but it's a very simple to follow illustration of the principle of PCR. And one thing I want you to pay attention to is the target DNA. So when you're watching this video, this bottom left is a little screenshot from that video. That green region is the area of interest, the gene that is being amplified. And right on either side of it is where the primers sit down. But I want you to realize that the target DNA is exactly what we're after, and we want lots of those double-stranded target DNA copies. We don't want any red on either side of that. And what you're going to notice is that as those cycle numbers increase, you get more and more, like exponentially more of these target DNA molecules, and you don't really increase the number of those ones with the red strands on any or either side, okay? So pay attention to that, because it's when you end up at the end with billions of these target DNA molecules that are all the same size, and just a few of these ones with red connected to them. That's why you can run a gel like what you see on the right, and what you get is a band of a very specific size, your target DNA. The vast majority of the DNA that is in the tube is one specific size, and it's the size of that target DNA. which is the area spanned by those two primers. It's exactly what you saw in Lab 3 when you calculated the flanking region and the number of repeats and how long the total fragment is, right? All of that is what's in between the primers, and that size is very characteristic. Okay, now where the real innovations have happened in your lifetime, okay, whether you were old enough to realize them or not, is in the sequencing of DNA. So let's see, yes, I was in graduate school before you were born, and I was sequencing DNA, okay? It was a very difficult and laborious process, and I might be able to sequence, let's see, in one night, staying up in the lab all night, I might get 250 nucleotides from 20 different snakes that I was working on for my dissertation, okay? Now, DNA sequencing has advanced so rapidly that I can, before I leave for the day, put something on a machine over in Natural Product Center, and when I come back in the morning, I have 400 million reads that are 75 bases long, okay? So hundreds and hundreds of millions of nucleotides, okay? Now, there are very, very different technologies for sequencing DNA, but it is this technology that we use not just to see how big your fragment is versus mine, but to read exactly what your nucleotides are. Every G, A, T, and C in order. And it is very helpful to use these to compare individuals. This is exactly what you were comparing with those Ebola virus sequences, okay? When you're comparing those individuals to one another, it's those differences that tell you something about which is most similar to which. For the most part, these technologies rely upon a polymerase that is actually stuck to the bottom of some flow cell, and you put a bunch of your DNA samples in there, and the DNA samples go through the polymerase that's stuck there, and every time a polymerase adds a nucleotide to a new strand that is complementary to what you put in, it releases a fluorescent molecule. Each nucleotide has a different color, and so there's just a camera sitting on top of this reading millions of different flashes every second as all of these different polymerases, millions of them, across this membrane that is about the size of your cell phone screen. And it is this that we use to read the DNA of organisms these days. I'm not going to get into the specific technologies because they're advancing so rapidly. There are so many different varieties. It's not really important at this point that you understand one technology versus another. Okay, now, what is the point of being able to manipulate this genetic information, being able to read this genetic information? Well, in making transgenic organisms, we can accomplish a great number of things. And one of the easiest examples that has been utilized in livestock is capitalizing upon those that produce milk, particularly those larger ones that produce milk like goats and sheep and cows, and putting a foreign gene, mixing it in or putting it nearby genes that are expressed in mammary glands for these mammals, okay? And so what you can do is you can get some foreign gene into an embryo of one of these milk-producing organisms, and once that embryo is born and grows up and produces offspring, the process that initiates milk production in that animal will stimulate the expression of this foreign gene that has been stuck into this animal, okay? So there are many examples of livestock, particularly goats and sheep, that produce milk that is incredibly rich in a human protein because we stuck it in that organism. And these are referred to quite often as biofactories. So think of a sheep that can, just by producing, say, half a gallon of milk a day, produce enough of this particular protein to supply 40,000 doses of that protein for a person that has a mutation that does not allow them to produce it themselves, just because we stuck this gene into a sheep that produces it. And it's easy to isolate a protein from milk. Now, you'll remember we spoke of the universality of the genetic code, and it's this same reason, this same transgenic activity that researchers are able to utilize to make these crazy fish that you see at the pet store. So these glowfish that you see at PetSmart and Walmart, they are all of these crazy colors because someone has stuck a gene from either a coral or an anemone or a jellyfish into these fish. These fish are able to transcribe and translate genes from very, very different organisms. Okay, a coral is about as far as you can get from an animal, okay, without being a plant or a microbe. So the fact that these fish can express these coral genes and produce these colors is quite remarkable. Now, this development was made primarily as an ecological test so that we could make fish that will turn colors if they're exposed to toxic water. So it's a pretty easy way to test water quality if you've just got a fish and you can put it in the water from a canal that you're testing or a river, and if it turns a bright color, it's been exposed to a pollutant. So that was the purpose behind it, but some ingenious investors received the license to also utilize it for beautiful pet fish. But this is a great example of transgenic organisms. Okay, now the last slide, or the last couple slides here, focus primarily on what is undoubtedly the most remarkable recent development in genetic technology, and that is CRISPR-Cas9. And so the way CRISPR works is it enables us to do something that had eluded scientists and doctors for generations. Since we knew that DNA was the material passed from one generation to the next, and we knew that mutations were the cause of different phenotypes, and some of those phenotypes were bad, right? Some of those mutations were deleterious, had negative consequences for the person who had them. It's really difficult to go in there and fix it. It was, until this point, almost impossible. Because while we are able to stick a gene for turning a fish red from a coral into that fish, if we can do that, why can't we stick a gene that fixes a genetic disease like cystic fibrosis into a human? Well, the reason is they didn't have to put that coral gene in a particular place in the fish genome. They just tried to throw it in there, and it randomly took in one of their embryos, and boom, that embryo, for some reason, expressed that gene that they put in a random spot in that enormous genome of that fish. What we need to do if we are going to genetically treat individuals for genetic mutations is we need to target the exact region where they have a mutation and erase it and replace it. And that is where CRISPR-Cas9 came to be. Okay, so this is derived from an immune system response that exists in bacteria. And so while scientists understood the role of CRISPR and Cas9, two separate things, within bacteria, what they didn't realize was the potential utility of this system. And so what this system enables you to do is one of two things. You can go to a region that is doing something that you don't want it to do anymore. Okay, so this is probably not something we would do in humans, but let's say we've identified a gene in a mouse. We're not really sure what that gene does. Well, we can use this pipeline on the left with CRISPR-Cas9, go in there and cut a portion of it, just a double-stranded cut to the DNA, and you know what happens there. Usually it will go find the homologous region if it's our cells, if you're diploid, homologous chromosome, and do that homologous chromosome replication repair, filling in that gap so it knows that nothing was lost. But with CRISPR-Cas9, you can ensure that it doesn't do that. It only just glues those ends back together, probably losing something, and you broke that gene. That's the intention of this NHEJ pipeline for the use of CRISPR-Cas9. The alternative is to replace a portion of a chromosome with something that you send in with CRISPR-Cas9, okay? And so in this case, we want to get this region stuck in there and replace the broken copy of the gene that this individual has, okay? This is where the real potential is for CRISPR. If you can go into someone that has a mutation that gives them cystic fibrosis or any number of genetic diseases, and you can simply cut it out and replace it with a functional copy of the gene, that's going to change the world, okay? And it already is. But it does lead to questions about, well, what if, we're about to have a kid and it looks like your kid's going to have brown eyes. Well, how much would it cost for you to fix that and make those green eyes? Okay, that can be done with this technology, all right, and a lot more. So as you learn about these new technologies, I want you to think critically about the potential implications. There's obviously a lot that we can do with it, but there's a lot of scary stuff that could be done. So there is a module that I'd like you to go through that explains CRISPR-Cas9. The textbook does an okay job of it. I feel like this is something that you kind of have to see and hear at the same time, and for that reason I'm providing you on the next slide the link to this module so that you can go through it and better understand exactly how this system works and how it can be used for the betterment of the world. Okay, so please do go through this. It's important that you understand CRISPR-Cas9. So this is the link, which you probably can't click on YouTube, but I will try to put a link in the comments below, and you can just pause this and type this as well. Okay, great.